University of Gothenburg in Sweden, who's a really awesome scientist doing a lot of great stuff on aquaponics. Thanks a lot for joining us.
Hi, yeah, and thanks for the invitation. So I'll just share my presentation screen there. And yeah, Angela gave a really great talk there. So I decided with this presentation, I'd kind of, is the screen visible? The presentation visible? Hopefully so. So yeah, I was gonna talk about microbial communities and aquaponics, and from a sort of a market perspective, because a lot of nutrient exchanges is, in a way, is really similar to a free market. My name is Victor Lobanov. I'm a PhD researcher at the University of Gothenburg in Sweden, as Steve mentioned. I'm not representing the University of Gothenburg as a cannabis growing institution because cannabis is illegal in Sweden, but just talking as a microbiologist and someone who's very interested in all types of aquaponic plant cultivation. So in this sort of talk, I'd like to introduce the rhizosphere, kind of what are some of the dynamics going on inside it in terms of energy storage via carbon and nitrogen, how other nutrients play a role in that exchange, how fungi play a role in rhizosphere composition, some microbial assemblages that would be found in aquaponic systems. And very briefly, I'll talk about some things that could be measured. This talk will be much more sort of scientific and research driven than as opposed to being a practical talk of things that you could implement. But I hope that people find it informative, because sometimes it's nice to sit back and look at what's kind of the state of the art for research. So I tried to do my best to review that. So looking at the rhizosphere is very interesting. Is a really interesting environment, because when we look at plants, we're thinking of the entire root structure, but the rhizosphere is really going on in this really narrow space. You can see in the bottom right corner, 200 to 5 micrometers, so 10 to the minus 6 meters from the edge of the root. And we're going to look at more in depth in that environment, but really all the action is happening there. And you can even zoom in more and see that it's not just the root, it's really like the hairs on the root where all the action is going on. And by action, I mean it's like the point where the plant's immune system and digestive system is interacting with the microbial community around it. And in a lot of ways, it's similar to our own gut microbiome. There are a lot of similarities one can draw there. And it's an environment that has all kingdoms represented. So you have archaea, you have bacteria, you have different kinds of protists, fungi, the plant itself, and also animals such as worms and nematodes. And when we refer to the living mass inside the rhizosphere, we call that the rhizobiome. And as far as like its function to the plants, the plant requires the rhizosphere for nutrient acquisition, water uptake. It's also its immune system, so abiotic and biotic stress resistance. And the microbial community itself, basically it lives off of the carbon that the plant sequesters. So that CO2 from the atmosphere, up to 90% of it goes into the rhizosphere. And the other 10% goes in towards actual plant construction and fruiting and such, which is quite extensive. And we're going to talk about rhizodeposition and how basically as the roots are growing, they're shedding a lot of cells, and most of that secreted carbon is in the form of these slowed off cells. And because of that, the rhizosphere is a really nutrient-rich environment compared to bulk soil. A lot of the research, of course, comes from soil-based systems. So where possible, I'll adapt that to water-based cultivation. But and we believe that most of the mechanisms going on are pretty similar from soil to water. And so in that first image I showed of the root hairs, the very tip of it is populated by these root border cells. And this is really like the point where the plant hair is penetrating into the soil. And this is where the most amount of nutrient exchange and such is going on. So this is, for example, the first line of defense. and trade, as I describe it, for the plant. And basically, as a function of the microbial community around the root hair at this point, the plant is going to be secreting different types of compounds. And this could be anything from exudates, which we'll talk about, secretions, so different types of secondary metabolites, and mucilages, which we'll also discuss in a moment. Those mucilages, for example, are similar to neutrophils in animals. So it's basically like a net that the plant can release, and it can use it to capture viruses and bacteria that would be potentially a threat to the plant. So it's really important that this microbial community is functioning well. And there's a lot of interesting things going on as to how exactly it's able to identify certain types of bacteria and viruses. But basically, for the microbial community, it makes sense to support the plant. For the plant, it makes sense to support that community. And when you remove one of those components, then you weaken the entire system. There's a lot of active research on this because we know that plants can identify certain pathogens, it means that it's possible to breed more efficient strains that are able to handle different kinds of pathogens. However, there's not really a lot of good strategies to do that right now, but that's something that the research is, there's the direction the research is going into. And for example, we could also find probiotics, different organisms that could help in that whole plant response. Looking at the composition of the rhizobiome, basically, if you were to take a random amount of soil that didn't have any roots in it, and then you were to look at that entire diversity, you would see that only about 7% of that community would also be found in the rhizosphere, meaning that there is a strong selective pressure by the plants to select for certain types of microorganisms that it wants to have. And that's just as true in aquaponics as in soil-based systems. And we believe that what's going on there is that there is a core root microbiome that forms. And then, of course, if we could somehow influence that or understand why exactly those microorganisms are attracted to that root and what kind of exchange is going on, if we could understand that, then we could really advance this. As that statement implies, we don't really understand a lot of what's going on there. But I'll basically just try to go through and share what we do know. So in terms of say in that kind of community dynamic, in terms of the ability to influence the community, plants have the greatest say. Filamentous fungi, so we'll talk about them later on in this presentation. They are also really adept at controlling the community, moving nutrients around and other microorganisms. And then after that, we have different kinds of protists, yeasts, other types of fungi, and then bacteria, and finally archaea, and which are all present, but individually do not have as much say as those other organisms. Microalgae in aquaponic systems is, they definitely are playing some role, but it's been hard to find good literature really digging into what role are they playing. I have my own theories, which we'll talk about at the very end of the presentation. And also I just want to mention that so the individual seeds have their own microbiome that the plant prepares for the next generation. It's kind of like a starter kit. And they are definitely used by the plant, especially as the seed is germinating. However, they do not predict the rhizobiome that will eventually form. So eventually, once the plant becomes a bit more mature, it starts selecting its own community anyways. And this graphic I've included just because I want to show the blue are enriched microorganisms in the rhizosphere, and the red are depleted microorganisms in the rhizosphere. Basically, this is what a lot of research papers look like, where they say, oh, we have all these different bacteria and archaea that we found in the sample, and we know that sometimes they're there, sometimes they're not there. And it's very, very difficult to say, okay, the presence of these organisms implies this type of metabolism, or when plant is this stage, it's going to be influencing the community in this way. We know that for example, some of the ways that the plant is able to influence the community is primarily through carbon and oxygen. So radial loss of oxygen is when oxygen is diffuses out of the roots into the rhizosphere. And you really don't need much oxygen to influence these communities because they're always on the brink of being anaerobic. And then, of course, as I mentioned, up to 90% of the carbon is going out through the roots. And this basically, it can set off a whole chain of things going on. There are different kinds of micro-niches where certain microorganisms may be anaerobic, but around them it's an aerobic environment, but they found that one little area where it's anaerobic. And basically, if there is a niche available, then some organism will occupy it. And this basically is really why it's so complicated to study these systems. So can we pinpoint how the rhizobiome benefits the plant? No, unfortunately. It's just, it's too diverse. There are too many compounds going on, being exchanged. Their half-life is really short. The concentrations for many of them are below what we're even able to detect. So we don't even know if we're able to detect everything. Sometimes they'll have a direct impact on, for example, there may be a specific organic acid that we know when it's expressed by the plant will enrich certain types of bacteria, but we don't know if it's happening directly or indirectly, how that's influencing nutrient uptake. It's this huge web, and so it's really, I think it's important to be aware of what's going on, but in the end, one of the reasons why I'm always so impressed with the farming community and why I was interested in cannabis aquaponics is that there's so many empirical studies that basic farmers are doing on their own, and is really one of the best things that we can do. And it's great to see trends form, and that, for example, having one type of cultivation system works better than another, and then to later go back in and understand what is going on to the extent that that's possible. So we're limited in our ability to have appropriate spatial resolution. We're talking, about micrometers of distance between the root and the microbial community and all these micro niches. So it's really small scale stuff going on. And then, as I mentioned, detection limits and our ability to even identify these compounds. And it's even harder to say what bacteria are responsible for what action. So bacteria like to do this thing called horizontal gene transfer, which I've shown in the graphics here. Basically, if you look on the bottom right, you can see horizontal gene transfer is literally when a bacteria or archaea, and sometimes a few fungi will do this too, they'll take a piece of their DNA and they'll just give it to another microorganism that they're not related to. And it's not a form of sexual reproduction. So it's very widespread, and it makes identifying the role of a certain bacterium very difficult, because you may identify the bacterium correctly, but maybe in the reference database, it's not associated with a certain type of metabolism that in this system it has because of horizontal gene transfer. Another thing that makes things difficult is that the amount of conserved DNA within a single species can be quite low. So for Escherichia coli, it's 40% of the genome is preserved across all subspecies, meaning that humans are more closely related to bananas than one strain of E. coli would be to another strain of E. coli. And for like Bacillus subtilis, it's around 20%, and it really can vary widely. So here, 10% for this example of Bacillus. And you can see this, this is kind of an example of what some of these research papers look like, where they will test a certain bacterial strain under different conditions to try to see what kind of phenotypes, so what types of genes they're expressing, how that differs across different strains. And it's extreme, the kind of diversity that you get in terms of preferences for different types of carbon sources or other nutrients. So now I want to talk about the good, the bad, and the ugly. So basically, we know that there are some organisms that are always good, and we call them probiotics or mutualists or plant growth promoting microorganisms. Some are universally bad, and those are obligatory pathogens. So if you think of a virus, for example, it needs to be a pathogen. It's the only way that it can survive. but the vast majority are ugly, and that they are opportunist or commensalist. Sometimes they're good, sometimes they don't really do anything. And the reason why I think that strategy works well is kind of along the lines of this quote from Lord of the Rings: "The closer we are to danger, the farther we are from harm. It's the last thing he'll expect." And basically, these are bacteria that live in the rhizosphere. Most of the time, they're not doing anything, but every now and again, if they see a good opportunity, they'll take it. So just to go through those three topics again, when we're referring to good bacteria, we're referring to functions such as being a bioprotectant, so that they actively suppress pathogens or predators such as nematodes, or that they can trigger plant defense responses. They could be biofertilizers, so that they enhance the uptake of nutrients. They could be biostimulants, so they produce compounds that will do something in plants that, for example, could improve the taste of the fruit, etc. And so lactic acid bacteria seem to be largely in this category, but it is very difficult to be precise about a lot of that, just because it's so diverse in terms of DNA, and there's a lot of research that still needs to be done. But this is when anyone is talking about probiotics, they're generally trying to refer to these types of organisms. And when we're thinking of the bad, these are basically R strategists, meaning that they try to reproduce really quickly, and they benefit from what I call high octane environments. So they have a type of carbon source, for example, like sugar, or it could be anything, some type of carbon source that they really like, some niche that they really like. And once they get that niche, they do everything to keep that environment that's suitable for them. And that usually and pretty much always comes at the cost of the community as a whole. So once they are established, they will actively try to proliferate and take over. And really, you can't negotiate with them. So if you're an obligatory pathogen, it's kind of, your cards are pretty clear. Opportunists, so they can be tolerated commensal organisms, meaning that there's, as I said, they're engaging with the rhizosphere. They're a benefit to plants even. But if that plant shows some weakness, then they can colonize and become a pathogen or virulent. They do that by having a good ability to adhere to host tissue, so they'll grow along the roots. They can form biofilms that protect them from plant defenses or from other attacks, basically. Or they can be good at evading the host immune system, or they can be really good at taking a certain nutrient that would be limiting otherwise. So usually iron is involved in opportunistic or pathogen strategies, because if you can control the iron, then you can control one of the most important nutrients in the rhizosphere that's relevant for photosynthesis in the plant. And so it's quite a coveted nutrient, also because its bioavailability is sometimes quite low. So yeah, the other thing I want to mention about opportunists is that they often are very closely related to pathogens and to plant growth promoting microorganisms. So it's not like lactic acid bacteria are always good. They usually, when they go rogue, it's just one species or one strain away from an organism that is really beneficial to the plant. And so that because of that, parasites can sometimes appear as commensalist, especially if the plant then adapts to accommodate for that type of niche in the rhizosphere. So sometimes the plant just gets used to having a certain type of opportunist, and then they create a new contract and a new agreement of living together. And yeah, so it's quite complicated. Now I'm going to try to go through a few different nutrients, and I hope this will be informative for you guys. So pretty much all carbon starts in the roots, or it could be naturally already present in carbon, or naturally already present in the soil. And it can go through a variety of pathways, but it always ends up as carbon dioxide. And so as far as the rhizosphere is concerned, we're interested in seeing how it's switching between available carbon in the soil into microbial biomass. That microbial biomass dies, or the root cells die, and then it goes back in the soil. And interesting enough, a lot of that is mediated by fungi. And fungi haven't really been explored to the extent that I would like them to be explored in aquaponic systems. But in soil system, it's very well known that they are a principal administrator of a lot of this carbon exchange. So they produce volatile organic carbons, which are like signaling molecules, and they can influence, they can promote or discourage bacterial proliferation. So we'll talk about that more, but just want to keep that out there, that as far as carbon cycling goes, they are extremely important, and we really don't have a clear idea of what they're, how present they are, and the extent in aquaponics or hydroponics that they're controlling things. And I thought this was a good visualization of how strongly carbon can influence a microbial community. This was a study in the Aquaculture Engineering Journal on biofilters, but they they noted that when they changed the amount of chemical oxygen demand, it's basically a proxy to measure the amount of carbon that's available in a sample. When they just by changing the amount of carbon going into the biofilter, they significantly influence the nitrification rate, meaning that they significantly change the nitrifying community. And so things like carbon, nitrogen, but I would say really those two elements first are extremely powerful as ways to mediate the microbial community. And I mentioned earlier that plants produce a lot of exudates. This paper that is cited here has a really great table where they list more specifically different types of exudates. So we know that they're really important for rhizosphere regulation. If anyone is interested in measuring the amount that they're being produced by their plants at different types in their life cycle, I think that there would be a lot of stuff that we can learn from that. But it's, as this list implies, it's a really diverse set of compounds. And not only that, but it also changes over time. So these are plants that were grown in a hydroponic setting for 9 weeks, and the profile of exudates that they were producing changed as a function of how far into the growth cycle they were. So there's, I mean, if anytime you see a signal like this, you know that we could measure something in that and know something about the state of the plant. It's communicating with us in some way, but we're just not able to really read this. And for example, one of the things making that complicated is that this study was done under sterile conditions, but of course we're not working under sterile conditions in an actual grow up. So how that can be used to predict real world scenarios is an entirely open question. And there are also some of those compounds, when they go into the water column or into soil, they become humic substances, which is a very general term for a variety of compounds formed through biotic and abiotic processes. Interesting enough, they are largely formed through the Maillard reaction, which is the same reaction that turns your bread brown when you cook it. I find that absolutely insane. But basically, it's kind of like those carbon molecules that are extruded by the plants are on a microbial level being heated up. There's no heat involved, but it's like the same chemical process. And that involves a lot of metals too. So that's why humic substances have been known to improve bioavailability of metals. But there's a lot that needs to be explored here. When we're trying to measure carbon, I thought that I would kind of show what are like the tools that we have available. So for an individual point in time, we can look at the total biological oxygen demand, PBOD. And from that, we can estimate the amount of bioavailable chemical oxygen demand, meaning that we can look at what type of carbon in a sample is available for microorganisms to use, and what type of carbon would take a long time for those microorganisms to break down. And that's really important because some bacteria, their generation may only be 10 or 20 minutes long. And so they don't have time to break down something that's really hard to break down. But like a fungus that lives, can live for days or years, it would have time to break that down. It would be more interesting for that fungus to do so. And so we can basically we can measure those two things, or we can just collect on a filter all the carbon that is in the water sample and put it in an oven. And depending on the temperature, if we're like drying it out, or if we're baking it at 450 degrees Celsius, we can also have an idea of the ash content or the total amount of solids, and that can be related to the amount of microbial proliferation, so the rate that the microbial community in that sample is growing. And we can also measure the amount of non-biodegradable soluble carbon. So there's different formula that kind of relate these sample metrics together. We can look at the amount of non-biodegradable particulate carbon, and then from those two, we can measure the amount of non-biodegradable carbon in the sample. So we can't really say what exactly is being secreted by the plant and in what concentration and what role it does, but at least we can kind of look at it from other perspectives. But as you can see, this is kind of limited and to the extent with which we can explain what is going on. And we also know that the amount of carbon dioxide in the surrounding the plant is also really important, and it's been associated with significant exudate profile shifts. So if you increase or decrease the amount of CO2, we know that it will have an effect, and that's about all we can say about that. So yeah, we hope to explore that further. But just to go over a few of the challenges that we're working with, and looking at an aquaponic setup, I made this diagram to show which areas have more or less carbon or oxygen or mineral nutrient accumulation. So the water source can have variable mineral nutrient accumulation depending on where it's coming from, which aquifer, what's your surrounding geology like. In the recirculating aquaculture system, you can have variable amounts of carbon depending on your flow rates and such. But once you go downstream of that, it's pretty straightforward. Like your solids, of course, you're going to have a lot of carbon, a lot of mineral nutrients, very low oxygen. So that's generally going to go and push the community in a certain way. Biofilter tends to have a lot more oxygen. You only need like 2 grams per liter to be an oxygen-rich environment, microbially speaking. And then the hydroponics bed, by that point, you tend not to have very much carbon, but you have other stuff going on. This was a paper out of Israel showing, trying to measure nitrogen and carbon throughout the cycle. And one thing that they showed pretty well here and then in other related literature is that in the biofilter, you tend to be losing a lot of nitrogen gas. And so there's a lot of, we can theoretically measure these things and get an idea of what that community is looking at, or what the community is trying to do. Archaea, for example, since they've been discovered in like 2005 or so, we now know that they are responsible, for example, for 1/3 of nitrification in the oceans, but we don't really have a lot of data on them, especially in controlled environment agriculture, because when you use the type of sequencing strategy for bacteria, it doesn't really capture a lot of archaea diversity. So we've been looking with the wrong set of tools. Switching to nitrogen now, I really like this graphic because it basically shows how many different pathways nitrogen has to become more oxidized or more reduced, and the different kinds of microbial communities and how they relate to each of those steps. So it's not just ammonia, nitrite, nitrate, unfortunately, or maybe fortunately. You have a lot of different, there are a lot of things that can be going on, starting with urea production by the fish, all the way to nitrogen fixation, which doesn't happen only in legumes, but also in all plants. But legumes just are really good at creating an environment for that. So about 20% of plant nitrogen is found in the rhizosphere on average, and that's usually in the form of those exudates that are coming out. So there could be like some proteins in there or cell wall components, because they're releasing a lot of cells as well. And bacterial communities are very sensitive to changes in nitrogen. So it's, I would say, generally best to keep those concentrations stable or rising or decreasing at a fairly regular and slow rate. And what the microbial community does for the plants in terms of nitrogen is basically it's involved in nitrogen fixation. It breaks down proteins, and then it will break down urea. So a lot of that is not only happening in the biofilter, but the beauty in aquaponics is that we're actually we're having a biofilter-like environment everywhere in the entire system because of the way that plants are interacting with the water column. So generally, plants like a nitrate and ammonia as their ammonium, sorry, as their preferred nitrogen sources, and they tend to not be that great at taking up nitrogen by itself. They really require the microbial community for that, and they have different uptake systems. I'm not going to go into too much detail here, but basically, they can take up more nitrate or ammonium depending on what's available, but they do like to have that combination. There's a huge open area of research as to how we can improve nitrogen uptake by improving nitrogen fixation. It's not too well explored right now, but basically the plant genotype plays a role, but also the microbial community, and we could potentially create probiotics for that. Can nitrogen uptake be improved on the plant side? We don't yet know enough about the uptake mechanisms to do that in terms of genetic engineering. However, you can select for certain types of plants that are better at taking nitrogen up. And one of the best predictors of that is having a lot of roots. So I've always felt that this kind of like root hatred in the hydroponic community was a little bit unwarranted. It may be that having a large root system will improve the fruiting bodies of the plant. So I think that's probably best to explore individually in different farms. As yeah, that microbial community is important. I'm just going to touch on phosphorus a bit. Basically, as far as the microbial communities are concerned, they can break down different types of phosphorus that they encounter. But as far as what's going on in the rhizosphere, they try to bring phosphorus into an orthophosphate form. So a lot of microbial processes related to phosphorus eventually will put in that form. So if you can measure amount of orthophosphate in your system, then you know what is directly bioavailable to the plants. So other nutrients, one thing that I've found in our own research is that we can add this trace mineral solution, and it pretty much always satisfies random trace mineral needs. But basically, different microbial communities require different trace nutrients, and even in really controlled laboratory settings, we have a lot of difficulty trying to identify whether it's like copper or magnesium or molybdenate, cobalt. But generally, when in doubt, we just add this Vishniac solution. So the microbial community is definitely really involved with it, but it's hard to say what are the ideal target concentrations. And looking at like ocean systems, there are basically 4 pathways that those trace nutrients are processed by microbial communities. So we know that organic matter is really important. We know that iron is like super important, and that all the types of iron hydroxide, so rust, that it can be formed in. Manganese, which tends to be quite absent in fish waste, but we know that it's very important for trace nutrients in plants and microbial systems. And then terragenous, which basically just means rare earth minerals, that they also play an important role. So something like aluminum, for example. I think that this would be a really great area for future research, but it's definitely, something like this is definitely happening in aquaponic systems, but we don't know enough about it. Sulfur, we know, is also really important. Here I want to point out that if you see on the left-hand side, 5% of sulfur in soil is in that top section, and then 95% is in that bottom section. So most sulfur is in an organic form. And primarily it's a sulfate that plants really like. So plants pretty much want to have that sulfur presented to them in a sulfate form. And they can also take it up in a slightly different form, but it's like really they prefer sulfate. And they can also take up sulfur dioxide through the leaves, which is convenient if you happen to live in an area that's quite polluted, because a lot of air pollution is in the form of, there's a lot of sulfur dioxide in air pollution. And we know that archaea, interesting enough, play a big role here. So they're not bacteria, but they're archaea are always these like very strange organisms that look a lot like bacteria, but have very funky metabolisms. And so they're doing something with sulfur. So reduced inorganic sulfur compounds, they are in, there would be things like H2S, which people may or may not know is highly toxic to humans, but also to fish. But for some reason, plants really like them, and they use them as a signaling molecule. So they are not bothered by that. And the microbial community will provide some amount of hydrogen sulfide. So they're in low concentrations, they're beneficial to plants, and they're involved in a lot of different stress pathways. It seems to be that it's a pretty profound system that the plant is able to use. So it'll be interesting to explore that further. But we don't have a lot of data measuring H2S and the rhizosphere or in the water column. So that's an area of future research. But this graphic, I think, summarizes things pretty well. That basically the plant is going to try to push things towards sulfate when it needs to increase the amount of sulfur in its tissue. And probably when that need is satisfied, then it's going to allow the microbial community to switch towards other types of sulfur compounds. Because things like sulfur and nitrogen for the microbial community are basically ways that they can store electrons. It's kind of like that free market concept that I mentioned in the title slide. All these things are in flux all the time, and it's basically you'll have one bacteria that's offering sulfate, and in exchange it wants to use that electron so that it can take up some form of carbon. And depending on the concentrations available to it, it's going to be, those compounds will have different values in that microbial system. And what's interesting is depending on the redox state, so whether there's more, on a chemical level, it's whether there's more electrons available or less. But basically, you can, as a proxy, you can look at like the amount of oxygen and dissolved oxygen in the water column. But in the rhizosphere, it's important to have both environments. So you don't want to blast the roots with oxygen, but you also don't want it to become entirely anaerobic. And it's by exploiting those two different environments of an oxidizing environment and reducing environment that plants and the microorganisms are able to create these 6 major signaling molecules. And so we've mentioned a few of them. I haven't mentioned the carbon monoxide, but basically it's they all have different and fairly complex pathways, but I just find that interesting. So if we would be able to measure those accurately in the rhizosphere, I think we'd be able to learn a lot about what the plant is trying to signal to the microbial community in terms of its nutritional needs. Where is the middle ground in all this? We don't have like a good number for a ratio. But we do know that, once again, fungi are really important here. So sulfur oxidizing fungi have 2 different pathways where they can manipulate the concentration of sulfur. And they are doing that with some sort of intention of influencing plant growth and root growth. But we don't know much about that. Iron basically switches between iron 3 up here and iron 2, and it is very redox dependent, and the microbial community plays a big role here. And I want to point out this scale on the right-hand side. So this is basically a scale showing the redox potential at a pH of 7, meaning that if you were to put iron, just the atomic iron, into, for example, a hydroponics grow bed at pH 7, then if the redox potential were 0.8, then there would be pretty much 50% iron 3 and 50% iron 2. And then depending on the type of chelating agent you're adding, you're going to be influencing the ideal redox environment. So at the bottom, you can see like iron EDTA requires like a slightly, like almost a reducing environment. So it's very, ideally there should be very little oxygen present at the same time that there's iron EDTA for that to be bioavailable to the plants. And if it were, so ferredoxin is used by a lot of microorganisms as a way to capture iron. And so at that point, they're going to be most active when there's a reducing environment. So basically what this is saying is the amount of oxygenation in your hydroponics grow bed is going to strongly influence all these different compounds independently of pH. So pH will influence metal bioavailability, but also the redox potential. We're actively exploring how different types of chelating agents can be more or less bioavailable to plants. Basically, it's also very interesting and very complex. So now I'm gonna, I hope that wasn't too overwhelming for different, looking at the chemical side of things. I just want to quickly look at fungi. Fungi colonization is ubiquitous, cosmopolitan, moderately non-selective, and occurs for pretty much all plants. The ones that we're mostly growing are angiosperms, and 80% of them must have a fungal community in order to survive. For gymnosperms, like pine trees and ferns, they always require a fungal community. And so fungi can basically, they can grow outside of the roots or inside the roots. And so we can refer to them as Endomycorrhizae if they grow inside the roots, or ecto if they grow outside. As far as we're concerned, the main thing to talk about here are Endomicroasia. Endomicroasia are also called arbuscular mycorrhizae. So I don't know why we have all these different names for them. You can call them AM, or you can call them Endomicroasia. It's all the same thing. Basically, they grow inside the cells of the roots, and then they coat the roots as well, forming what's called a fungal sheath. And for their services, they usually will take up 20 to 40% of all that carbon that is being released by the plant. So it's kind of a little mafia-esque. The plant really doesn't have a choice. But what they do for the plant is a wide range of services related to basically water retention, nutrient uptake, plant defense. They will attract other microorganisms and move them around. And they do often, I would say, Endomycorrhizae, so the AM fungi, they always have the plant's needs in mind, and they tend to be very selective about what kind of like types of plants that they prefer. And one of the reasons why I am fairly confident that they are active in aquaponic systems is because of a particular protein called hydrophobins, which I used to research beforehand. And basically, hydrophobins, they are used by the fungi to grow on surfaces. So it could be a water-air surface, it could be water-solid surface, but basically they're very happy with water-dominant environments. And because they are going to be looking for fungi, or their fungi will be looking for plants to colonize, and because they're able to grow quite fine in water-dominant environments, I would bet that if you were to look for them in aquaponics and hydroponics even, you would find a huge amount of diversity. However, it's kind of difficult to pinpoint what types of fungi to add as a probiotic, if anyone would be interested in that. And that's because they reproduce in a lot of strange ways, and it's hard to be really specific about the strain. So, for example, people talk about trichoderma. Trichoderma is a great, most trichoderma species will form symbiotic relationships with plants, but sometimes they can be opportunists. As we talked about earlier, it's kind of hard to tell the difference. And so these are some pictures of what those AM fungi look like. And you can see that they will grow towards the root and then grow inside the root and then reproduce all around the root. And they help with something, for example, salinity stress. So if that root, salinity stress is the same thing as drought as far as the plant is concerned. So if that root is drying out, then fungi are going to be able to tell the plant how to respond to that faster than the plant would be able to do it on its own. So there are a lot of, I don't really have time to go into much detail on this, but basically there are a lot of compounds that we've identified at the bottom of the page here as indicative of salinity stress and drought. And we know that the fungi are able to influence it. But it's hard to say, like, okay, if you add, if you measure the pH, then that means that it's like stressed in this way. But that would be the place to look if you wanted to find out information on how the plant is responding to salinity or drought stress. So yeah, just to summarize the whole discussion on the rhizosphere community, that community does not want the plant to die. And while some beneficial microorganisms have been identified and could be used as probiotics, there's still a lot that we're still having trouble discerning between opportunist and plant growth promoting bacteria. In the water column, this entire thing is also going on. And there are a lot of different names for microbial assemblages. Some of them are funnier than others, in my opinion. Biofloc is often used when you talk about like shrimp cultivation or sort of like dirtier water cultivation. If you're an environmental microbiologist studying the ocean, then you call it marine snow. If you're from University of Massachusetts and you're looking at the Sissiput salt marsh, then you call them pink or green berries, depending on whether they're sulfur dominated, so the bottom left photo, or algae dominated, the bottom center photo. And the composition of these communities is extremely diverse. For example, so when we look at freshwater microbial communities, we've been able to generally associate certain families with certain roles. So this is kind of a rough overview of what some of those look like. This is pretty much what we're reliably able to do right now. Say that, like, generally when you have these types of bacteria, they're probably going to be doing this kind of metabolism. And so one thing that we would really like to do is create something called designer biofloc, where basically, ideally, we would like to pick strains that are able to do certain functions for the plant and for fish. And I think in the next 10 years, we'll probably have this at least done to some extent. But really, the sky is the limit. And we do think that we need to go beyond just looking at bacteria, start looking at protists and other eukaryotes such as slime molds. Slime molds are not fungi, but fungi also, of course, would be involved here. Such fungi such as yeast, and maybe as well like filamentous fungi or the AM fungi that I was mentioning earlier with the root systems. And these microbial assemblages, they start off just free-flowing microbial cells. They start producing sort of sticky substance called EPS, exopolymeric substances. Like, it's kind of basically like polysaccharide glue. They start communicating with each other, which we call quorum sensing. These granules, as they form, they eventually get a dead space inside. And they also get different aeration zones. So the edge of these kind of microbial assemblages or granules tends to be aerobic. And so you get, these are ammonia oxidizing prokaryotes, AOP, nitrogen or nitrite oxidizing bacteria. And then you get some heterotrophic bacteria. And as you can see the scale here, we're talking about a really tiny sliver of these granules. And then the bottom part, you're going to have different types of nitrogen metabolizing bacteria, such as Comomox, Anamox, denitrification going on. And then in the very center, you're going to have a dead zone. And it's there's a lot of potential with the free-floating communities, but then also biofilms. While biofilms are often associated with biofouling, it's really a simplification because 99% of all microbial life is organized in biofilms. And they're extremely important. And theoretically, you could have a system where your biofilm community, which you're influencing by manipulating certain nutrients in the water column, is protecting the system as a whole. So meaning that the pathogens would not be able to colonize the system because of the biofilm and the microbial assemblages being organized in a certain way. Biofilms are resistant to water flow. They are resistant to antimicrobial agents as produced by pathogens through the extracellular polymeric substances, the EPS that I just mentioned. And they have a lot of micro niches, so relating to the strata in the biofilm. So as they are forming, this is kind of similar to the previous two slides, you're selecting for different types of microorganisms, and it's that diversity that's quite powerful. So we're really interested in understanding how do we get good biofilm and how do we prevent biofouling? Because biofouling seems to happen when the environment is unstable. And as I mentioned in that pathogen slide at the beginning of the presentation, when you have that niche that a pathogen likes, that pathogen will start colonizing it and then make other niches around it more like the niche that it prefers. So it will force its lifestyle onto the community. We know that carbon is really important here. So the accumulation of carbon could potentially lead to this kind of instability. And there's generally right now we can say that the best strategy is to limit the excessive accumulation of carbon, but maybe in the future we'll be able to have a better answer. In terms of what we're able to measure, I don't know how practical, and I don't think that a lot of this is practical for individual farmers, but I hope that we'll be able to integrate more community-based research projects where farmers could measure the total suspended solids, which is just filtering the water column onto a filter, sending it to us to be analyzed, measuring the amounts of different types of nitrogenous species, and looking at various profiles of the amount of nutrients in the water column or inside the plant through plant sap analysis. And just to, in the end, when we have enough data, we can make better models. We'll be able to understand more of these biotic and abiotic interactions, which currently are way too diverse and dynamic to predict. But we are learning a lot, and you know, got to keep in mind that like it was only in the 1980s, 1990s that we started sequencing, period. And a lot of the data that we've collected is only like in the past 10 years, and we're still in the process of integrating different data types. And it's just such a new field in the grand scheme of things. So we just need more time. And thank you very much for your attention. Feel free, if I will try to answer questions. I kind of went a little bit longer than I had hoped. If I don't get around to answering an individual question, or if you think of a question later on, feel free to send me an email. I enjoy talking about this, and I tried to not go into too much detail, but at the same time, I wanted to go into what we know as far as the scientific side of things. Yeah, so I'm gonna stop the sharing and then just go back to normal video.
Sure.
But
we had a question. That was an amazing presentation, by the way. Thank you so much.
Thank you.
We had a question from Chad about do biofilms present a hazard on soil surface when built up to extreme numbers in compost tea brewing processes?
Well, as I mentioned, that the carbon, the amount of carbon in the system can trigger these sort of niches that are preferred by pathogens. So there is that potential that if you push that physiochemical environment in a certain way, you can inadvertently create a niche that you don't want to create. In the end, stability is the best. So whatever is working whatever is working well and preventing fluctuations is probably going to be the most stable and therefore the safest system. It's something that the plants and fish will be able to get used to, and then they'll basically always be looking out for each other, kind of like how you know, if you imagine in like the African savanna, all the herbivores tend to look out for predators together. It's kind of the same thing going on a microbial level.
Have you done any microbial inoculations and testing around that, or are you strictly doing mostly observational stuff? And if you have done inoculations, what did you try and what kind of results did you find?
Well, a couple years ago, we were looking at inoculating phosphate solubilizing bacteria in a hydroponic system. And I had bought, in the Netherlands, I bought some sort of commercial probiotic that had claimed to have these phosphate solubilizing bacteria. Interesting enough, when we sequenced the probiotic that we had purchased, the advertised bacteria were not present. And that's a common problem in a lot of probiotics in the European Union and in the United States, that there's not the same kind of regulation. We haven't really done a lot of work looking at individual probiotics because we've been trying to understand the mechanisms of how probiotics work. One thing that I would really say about probiotics is, imagine like, you know, yogurt and kefir are generally considered to be quite good probiotics for humans. And when you grow yogurt, or when you're growing a probiotic for plant inoculation, that environment that it's being grown in tends to be quite different than the end environment that you're putting it in. So when you drink yogurt, those bacteria are really not able to colonize your gut microbiome. They enter, they play some sort of role, and we don't really, like for yogurt, we don't have like a very clear understanding of what is going on. And observationally, we see that it's good, that like it's healthy for us. And so I think that with a lot of probiotic application, I think it may be difficult to say what is being applied and what is staying, and what is being applied and triggering some sort of effect. So right now, what we're doing is mostly observational because we're trying to understand kind of how useful are probiotics and where is the right, you know, time and place. You know, you want to be applying something that the plant will accept as well. So maybe it's a great probiotic based on lab studies, but maybe the plant doesn't need that at that moment or doesn't like it. Gives me a headache sometimes how complicated all these interactions are.
Have you seen any negative observations with particular bacteria or other microorganisms in this realm?
Yeah, so because of the genetic similarity between pathogens, opportunists, and plant growth promoting microorganisms, it can be very easy to grow a certain culture that ends up becoming an opportunist. Or, and Angela had mentioned this in her presentation, if you apply too much of something, then that can also ruin things. For example, each microorganism is going to require certain things to survive. It's certain foods, and if there's a scarcity of those foods, then you can start getting some type of competition that you may not want. It's yeah, so it can definitely there's a lot of really great work in terms of the Korean natural farming, regenerative agriculture, and what I like about a lot of those strategies is that they've been developed over many different biomes. So, you know, it's kind of being done a little bit all over the world. And there's strategies that tend to err on the conservative side of inoculation. So a lot of things are quite diluted, because in the end, if that bacteria is really a good plant growth promoting microorganism, then the plant will promote its growth as well. So it just needs to be there in enough number that it can survive. And then if the plant wants it there, and if the fungi want it there, then they will promote it. So you don't need to add huge amounts.
Awesome. Well, it's definitely good to know. How can people find out more about you and the work that you're doing?
Well, I guess the best way would just be through different conferences and stuff. We're really just focusing on the research side. So, and because we're a public institution, we don't have like a website or anything besides the university website. But we'll be putting out articles and talking in conferences. So yeah, hopefully having more talks and such too.
Do you want to put your email back up on the screen real quick so people can email you if they have a question?
Yeah, so I'll go like this. Yeah, so victor.lobanov@gmail.com.
Oh, there we go. Audio.
Yeah, that's Joe's computer. Audio. All right, I will log off.
Thank you so much.
Thank you.
It's a great